Assalamu alaikum, dear students. I am Muhammad Rauf, lecturer, Department of Zoology, Cust. Welcome to the course Principles in Animal Life 2. This is lecture number 28. The topic is Translation in Eukaryotes. Discussing agenda for the topic is building a polypeptide, completing and targeting the functional protein, making multiple polypeptides in bacteria and eukaryotes. Lecture outcome. After watching and listening this lecture, students will know how the information stored in mRNA is used for the synthesis of a polypeptide chain. This is the second video of the topic, and we will recall what we studied in the first video. So translation is the RNA directed synthesis of a polypeptide. Genetic information flows from mRNA to proteins through the process of translation. You should recall it from the lecture 13, where we studied the central dogma of molecular biology, or the central dogma of life, which was stated by Francis Crick in 1956. Francis Crick stated that the transfer of genetic information in a biological system flows from DNA to RNA to proteins. DNA carry instructions for the synthesis of enzymes, proteins, and for the production of other types of RNAs apart from messenger RNA, and then only the messenger RNA, which carries a message for the synthesis of a polypeptide chain, is translated. Other types of RNAs copied from DNA are not translated. They are slightly modified after transcription, and then they becomes functional. So translation is the synthesis of a polypeptide using information in the mRNA. On the right side of your screen, you can see a process of translation going on. This is the messenger RNA which arrived from the nucleus. This is the ribosome, which are universal organelles, which are protein workbenches, which are protein factories. Ribosomes are composed of two subunits. This one is the larger one, this one is the smaller one. Ribosomes has three slots, E, P, A, exit site, where the tRNA leaves a peptidal site, where the tRNA holds the growing polypeptide chain, and A site, where the incoming tRNA is located. So now coming to the actual topic of that video, building a polypeptide. So there are three stages in which a polypeptide is synthesized. Those three stages are also there, means these processes are divided into these three stages for convenience in learning. So these three stages are initiation, elongation, and termination. All these three stages require protein factors that aid in the translation process, and energy is provided by hydrolysis of guanosine triphosphate, which is equivalent to ATP. Ribosome association and initiation of translation. So this is the first stage. The initiation stage of translation brings together messenger RNA, a transfer RNA with the first amino acid and the two ribosomal subunits. So when these three components join together, this process is known as initiation. What are the three components? The messenger RNA, which arrived from the nucleus, talking about eukaryotes, the transfer RNA, which was already located in the cytoplasm, and the ribosomal subunits, which were synthesized in the nucleolus, and then they found their way to the cytoplasm. So the aggregation of these three components results in initiation of translation. A small ribosomal subunit binds with mRNA and a special initiator tRNA. The very first tRNA is the initiator tRNA. Then the small subunit moves along the mRNA until it reaches the start codon. So first an association is established, then the codons needs to be at the proper sites on the ribosome. The ribosomes have 3 slots, so the codon which is to be translated into an amino acid needs to be at a correct location, a correct site on the ribosome. Here you can see this is the initiation process. This is the ribosome, the messenger RNA. Now don't get confused by the fact that why the translation starts from here and why not from here. Remember, we studied during the process of transcription that on both ends of the messenger RNA there are certain sequences which are not translated, and these are called UTRs. UTRs means untranslated regions. So there are some sequences which will not be translated. Similarly, there are sequences which will not be translated. But why they were attached? They were attached with the messenger RNA for the purpose to elongate the life of messenger RNA and for attachment of messenger RNA with ribosomes correctly. That was their function. So here is the first codon which will be translated into an amino acid, and the first codon is always methionine, the first codon in any messenger RNA. Anywhere in prokaryotes or eukaryotes which is translated is methionine, and AUG, this is the sequence located on messenger RNA, and it forms complementary association with anticodon present on the anticodon loop of the transfer RNA. Once the complementary association is established, then this transfer RNA is in a position to leave that particular amino acid, and then the process is repeated. So this process is energy consuming, and 1 GTP is lost during that process. Energy expenditure occurs in the first and third step. Codon recognition requires hydrolysis of one molecule of GTP, which increases the accuracy and efficiency of this step. This recognition of the codon, it is an energy consuming process. One more GTP is hydrolyzed to provide energy for the translocation step. Now, as I already stated in the previous video, that the very first transfer RNA will enter P site, and after that all other transfer RNAs will enter A site one by one. So now in the next slide we will see where this slot will also be filled by an incoming second transfer RNA. So what will happen when the two slots are filled? This slots needs to be evacuated so that a third transfer RNA can arrive, and it will only happen when this ribosome shifts its position. How much? Equivalent to 3 codons. Here here you can see this is the first stage where initiator tRNA arrived at P site, and the second tRNA arrived at A site. Now both these slots are occupied by tRNAs. So what will happen? This ribosome needs to shift one position in that direction. One position means equivalent to 1 codon or 3 bases, and now it has occurred, it has taken place. Here you can see the tRNA which was present at P position shifted to E, the tRNA which was present at A position shifted to P, and the A site is free for another incoming tRNA. So this is also an energy consuming process, translocation, where the ribosome shifts its position along the messenger RNA. And there are certain proteins which are called elongation factors or EFs, and this process is aided by these elongation factors. Then there are release factors or RFs which are responsible for the termination of translation. Here is the termination of translation. In the previous slide, this was about elongation, the second stage of translation. So this process will continue till the arrival of a stop codon. The same process will continue, no matter how long the polypeptide chain to be formed is And remember that the sequence of amino acids in a polypeptide is related with the sequence of codons in a messenger RNA. The type of sequences, the type of codons which are present in messenger RNA will be translated in the same way in the amino acid form. So the sequence of amino acids in a polypeptide corresponds to the sequence of amino acids in a codon messenger RNA. This is the termination of translation. Whenever one of these stop codons is encountered, then there is no amino acid which is represented by these stop codons. So release factors will arrive here, and these release factors will end in the dissociation of this messenger RNA from the ribosomes, and it is also an energy consuming process. Now what happens once a polypeptide chain is synthesized? Remember that immediately after synthesis, a polypeptide chain is unable to perform its function. It must undergo certain modifications called post-translational modifications of proteins, or PTMs, in which different functional groups are attached with the polypeptide, and if the protein is a complex one, a complicated one, recall the tertiary and quaternary structure of proteins, then there is the attachment of another polypeptide with that polypeptide. For example, an enzyme is present in the liver which is composed of 20 polypeptides, and each polypeptide is 2000 amino acids long, so some of the proteins can be humongous. Often translation is not sufficient to make a protein functional. Polypeptide chains are modified after translation or targeted to specific sites in the cell. Protein folding and PTM. During synthesis, a polypeptide chain simultaneously coils and folds into 3D shape. Now remember, I think I have already stated that in biology there is an important rule that structure determines the function, and it is extremely important for proteins and enzymes. You already know that the enzymes have specific binding sites for their substrates, so the synthesis of a polypeptide is not enough. This polypeptide must fold, must twist, and must come into a correct shape so that it can perform its function efficiently. Protein may also require PTM before doing their jobs. Targeting polypeptide to specific location. Now let's suppose the process of modification, post-translational modification is over. Now the proteins, the synthesized protein needs to be transferred to the correct location. So how this process happens? Two populations of ribosomes are evident in cells: free ribosomes and bound ribosomes. Now a cell produces two types of proteins. One type of proteins perform their function within the cell, inside the same cell. The other types of proteins are transferred to other locations outside the cell. And these two types of proteins are synthesized on two different ribosomes. Recall that ribosomes are of two types, one that are freely swimming in the cytoplasm, the others are attached ribosomes which are bound to the rough endoplasmic reticulum. So free ribosomes mostly synthesize proteins that functions in the cytosol. Means the free ribosomes which are there in the cytoplasm, they will synthesize those proteins which will function within the same cell. Bound ribosomes make proteins of the endomembrane system and proteins that are secreted from the cell. What is endomembrane system? All the membranes inside the cell forms a system which is called endomembrane system. So the endomembrane system, recall the structure of plasma membrane, which is a fluid mosaic where hundreds of proteins of different types are embedded in the bilipid layer. So from where these proteins are synthesized, they are synthesized on the bound ribosomes, and the proteins which will leave the cell and go to other destinations to perform their function will also be translated on the bound ribosomes. Polypeptide synthesis always begins in the cytosol. Synthesis finishes in the cytosol unless the polypeptide signals the ribosome to attach to the endoplasmic reticulum. Polypeptide destined for the ER or for secretion are marked by a signal peptide. So if a polypeptide that is destined to go to the endoplasmic reticulum or to outside the cell, they are directed towards these destinations by signal peptide. And these signal peptide, they bind to sequences which are located on that messenger RNA. Signal recognition particles binds to the signal peptide. Making multiple polypeptides in bacteria and eukaryotes. Suppose a gene was transcribed, messenger RNA was synthesized, and then this messenger RNA was translated, but the cell is in need of more proteins from that particular gene. So what will happen in that case? That after a single translation event, the messenger RNA will not be degraded, but more and more ribosomes will come and they will attach with that messenger RNA, and there will be a structure where several ribosomes will be attached with the same messenger RNA, and that structure is termed as polyribosome, and this is also a part of gene regulation. Why this happens? Because the organism is in need of more proteins from that particular RNA segment. So instead of transcribing another messenger RNA, why not use the messenger RNA already synthesized multiple times and produce multiple polypeptides from it by attaching several ribosomes to it. So you can see here several ribosomes are attached, and the same process which is discussed earlier occurs at each site, and several polypeptides are produced. So this is the summary of the whole process. Transcription, where messenger RNA is synthesized from DNA. This messenger RNA undergoes processing. There is attachment of poly A tail and cape at the 5' end. Then there is the removal of exons, and after messenger RNA reaches maturity, then it finds its way to the cytoplasm through the nuclear pores, and in the cytoplasm they are attached with the ribosomes. Ribosomes have three sites: E, P, A. E site where tRNA leaves, A site where the tRNA holds the grooving polypeptide, and the A site where the incoming transfer RNA attaches. So there is a slight mistake in that diagram. This AUG must be located here instead of that position. So this is the overall structure and overall process of transcription and translation. Thank you very much. Allah Hafiz.